This is for bacterial enumeration, and we use a stain called DAPI, so we typically call it making DAPI slides. And it's a way of quantifying the bacteria in the seawater. So to start the process, we first need to thaw the samples if they're frozen and get them up to room temperature, because the formaldehyde that we fix it with is a bit like olive oil, so it kind of has that funny kind of almost precipitate when it gets cold. The other thing that we need to do is we need to stain the filters. The reason we have to stain the filters is because looking at a bright thing on a white background, you don't have as much contrast. So we stain it with a stain called Ergon Black, which is a carpet stain, and it just stains the filters so that they're a nice grey colour, and it gives better contrast when we're looking at them on the microscope. So usually those are the first two things that you do. You thaw your samples and then you stain your filters. Then when you start, we have towers with a specific diameter. So there's 25 millimeter towers that we buy, but the inside of the tower is smaller. So it's usually around 17, 18 millimeters. So we have to predetermine that, so we measure that ahead of time. And we have bases, which are just glass bases, and these are clipped together with a clip. But in between there, we need to put a backing filter, and the backing filter is a 0.8 micron filter that basically helps evenness of distribution on the filter. And then the actual filter that we use is a 0.2 micron, which is small enough to collect the bacteria on top of the filter. We use a polycarbonate filter so the bacteria actually sit on top of the filter, they don't go into the filter. So then we can do an accurate count. So we put the backing filter on, the stained filter, we clasp the filter towers together, and then we filter anywhere from 1 mil to 40 mils. So for example, the Devil's Hole project, there's a lot of bacteria at 24 meters, so we back down to 1 mil. Whereas the bat site in the deep ocean, there's not very many bacteria, so we have to increase it to 40 mils. Then once all the water is filtered through, we then put the DAPI stain on, and this is a 5 nanogram per microliter or a 5 microgram per mil solution that we put on the filter. We stain for 3 minutes, we vacuum it through, then we can mount the slides. So the students have been mounting the slides. We use a high viscosity oil, we smear it on the slide, filters stick to it. They have to remember to keep the filter upright, because if they turn it upside down you don't see the bacteria. And then we put a cover slip on with the high viscosity oil, and you've got to press down. Another thing that the students tend to do is they put 2 cover slips on, so when you're looking on the microscope you can't see anything, but then it's quite easy to just take the top cover slip off. And then once all the slides are made, we just go into the microscope room and we count them manually. There's a grid in the eyepiece of the microscope, and we count a certain number of rows until we get to about 16 cells.